Good morning, Professor Ventura, and thank you for finding the time to have this short interview in the context of machine learning for non-globular protein. In our cost action, we usually interview scientists that are part of our group. And actually, we were wondering what is your idea or what are your ideas about integrating different approaches, considering that your lab is indeed integrating the computational approach, but also the experimental one.
So thanks a lot, for the question. Thanks a lot for the interview. Happy to be here in this consortium. Now, the idea, there's a story on why we mix computation and experiments, and essentially is this because I am by formation experimentalist, but then during my postdoc I made computation, right? And then it arrives one time, you have to go back to your lab, set up your lab in your institution, right? And then we were in Spain. So at that time, we have like the decision that we don't have enough infrastructure to be super good on experiments, even though we have good ideas. And then also the same happens for the computational resources we have at that time. So the idea is that, okay, even we will not be super good in one thing or super good in the other, we thought that at the interface we can have a niche and be there. And this is, when the river goes to the sea, this is the place where you fish the more. And we thought on that, and I think we have been implementing this with quite success. And the idea is always the same. So what we begin is with computations. So we make predictions. We work on protein aggregation, and either in the previous times which region of the protein are important for the behavior it has, and more recently we are trying to develop drugs. So how these drugs interact with the regions which are important for aggregation, or with the structure, or with the toxic species. And then it comes the idea that then the experimentalists test them. So the experimentalists are side by side to the computational guys, and then they have decided to be like that. So in a way, say the computational guys, do you want to be with the noise of experimental lab? And they say, yes, we want to be together and this is, so we make the experiments. In many cases they fail, but then you retroalimент and this is a cycle which I think is quite productive. And in 2-3 rounds you can have something which is relevant and is validated experimentally, and then in the papers they go together. And something which is, I think, very nice is that we like a lot of collaborations, but then computational people and experimentalists, they speak different languages, and then you take significant time in the meetings for explaining what you are doing. And these guys, they understand each other because essentially they are going for lunch together and they talk on the experiment. So this is a success. And I think in more general terms, not only in the context of my lab, this is something that should be done. So knowing a little bit of the two things give you fast, and then if you need more computational power or you need more experimental settings, you can always go in a collaboration.
Mm-hmm. Nice. You were talking about misfolding proteins and aggregating proteins, which we refer to as IDP. And actually, there are certain groups that are focusing on a specific protein and a specific neurodegenerative disease. But then there are other groups, which yours seems to be one of those, that focus more on the general aspects, on the misfolding in general, which is something that it might be an advantage because it consider a broader aspect of the diseases in general. What do you think about?
Yeah, this is, I think, a personal decision of how do you want to run your research, okay? It's good to be a specialist. There is no doubt about that, and being like a specialist in your disease, whatever. But I think, many of these neurological disorders, they share molecular mechanisms, especially misfolding, aggregation, and the toxicity of the aggregated species. And I think, by understanding, you can always, touch one more than the others, but what is now clear in the field is that what is learned from one of the diseases, essentially the mistakes that are learned from one of the diseases should be avoided to be translated to the other ones. And in one of these cases, famous cases, is the immunotherapy for neurodegenerative disorders. So essentially, there have been many failures in Alzheimer's disease and people working, let's say, in Parkinson, has been following what's going on in Alzheimer's disease. And now we have, like, for the first time, something that seems to work in Alzheimer's because it targets what is called the soluble aggregates. And then immediately can be translated to Parkinson's disease. And this is something we are doing and many other labs are doing. So as long as you have general mechanisms, then you can decide. It should not be a general drug. It will be a drug for each specific one, but it will target the same kind of species. I think this is an advantage. And another thing is that you cannot be a specialist of everything, right? And neurodegenerative disorders are so complex that you need multidisciplinary work. I mean, it's not enough being good in bioinformatics, not good being in biophysics or in animal models. You have to integrate a team that works together, in most of the cases also a company that is providing support, because at the end of the day what you want to do is to bring it to the patients, and then you need an integrative team. So focusing on a single disease is good, but you will, I think, arrive until a point in when you will need the help of others, and most likely these others should have like a more broad knowledge of the mechanism of action. And this is the way we think. So aggregation is something common to these ones, and by learning how this process behaves at the molecular level, I think you can address different disorders, and it's clear. So it's not that the community is inventing something for Parkinson's, something for Alzheimer's, something for Huntington. We're essentially using the same principles to develop drugs that, of course, because there is some sequence specificity there, should be different. But at the end of the day, well, in our case, what we want is that the protein didn't misfold in aggregate and they keep in the native form. And this is common to all of them.
Mm-hmm. You were talking about computational approaches and bioinformatics in general, and now you have been into the field for a while, and you have seen maybe an evolution of the vocabulary that is being used to machine learning approaches, and whatever was called bioinformatics until 3-4 years ago, it's now referred to as AI or artificial intelligence in general. What do you think? Should we still be careful about distinguishing those two macro fields?
Yeah, absolutely agree what we are proposing. I mean, I think there is like kind of rebranding of the topic. So essentially, many of us, and include myself, try to put AI because when you think you are going to have more chances to be funded, right? Because it's a trend also from the governments to push artificial intelligence, so you want to put it everywhere. But I think still, I mean, bioinformatics is the basics and the foundation of computational biology. So we need to understand how sequences are processed. We need to understand how structural models are made. We need to know how statistical programs work. Because then we have the risk, and this is something that's happening in the community, that we have kind of blind predictions, right? So, which are super good, but if we put in, and we have been discussing in this meeting, AlphaFold 2, AlphaFold 3, at the end of the day, we cannot distinguish between them because we don't know what is behind that. So it's kind of a black box. And I think we have to put this still a lot of effort in the bioinformatics part, because this is what is going to give us the interpretation of things that, of course, artificial intelligence are going to do, like processing large data sets that you cannot do with other methods. But you're going to need this, like, say, old-fashioned tools to interpret what is happening at the single protein level, at the family level, at the organism level. And I think, I mean, we should not confuse the terms. So artificial intelligence so far is something, and then, but still we need the bioinformatics. And of course, all of us are trying to do both things because the power of artificial intelligence is super high. But I think what we need now is moving from what we are doing many of us is like, using artificial intelligence for specific projects to develop specific artificial intelligence tools for our own projects, taking into account the knowledge we have learned during the last 20 years in these topics, and not like, because otherwise we are going to be becoming users and we are going to lose, the power. So we have to become developers. For for the specific problems we address. And this is something we are trying to do with aggregation, which is our own topic, right? To try to use basic principles to inform artificial intelligence tools or algorithms to look for things that make sense and we can somehow interpret.
Actually, the amount of information that were generated in the last, let's say, 15 years is much higher than, for example, compared to the years before. And something that seems very, very prominent in your way to talk, also at conferences or from your social media profile, is the effort to communicate both with the scientific public, but also with the non-scientific public. So I think not every scientist is done for this kind of mission, like communicating science in general. But I think it's also important that some of the people involved in doing research are also spreading what they know, and they try to sort of translate those concepts in maybe simpler terms or understandable terms also for people that need to take decisions. So the policymakers, but also people's involved with, for example, with the disease or the caretakers. So I would like to hear more about that.
Yeah, I mean, I fully agree with what the words you are saying. So essentially, I mean, for me, communicating is not like a luxury. It's a mandatory for us. I mean, we are the guys that are making the science. In many cases, on the large majority of cases, we are doing this with public money. So money that we collect either from the government or for specific foundations. And we have to communicate that what we are making. That's, I think, is for us, an obligation if you want to see. It's something that is necessary. And this is something I try to translate to my lab, but also, as I know, I'm directing now a new institute, also to my institute. So we should communicate in two levels. One level is to scientists, right? So it's better that you communicate your science, that let the science to be communicated by others, because you are giving the right message. The message which is transparent, is clear, and in this level is scientifically sound. So you don't overstate things, and I think this is important. But the other level is also at the level of the public. So essentially, we have like this disconnection between the society and the science. You don't see much science in the media, or at least in the TV or in the radio. And this is because we don't connect with people with the words that they can understand. So this is what, it's inherent to me. I try to communicate this to the people in the simplest manners, but in the most accurate manner it is possible. And this is especially important for people like that has diseases in our area, neurodegenerative diseases, right? So they are waiting for a cure. But you have to be realistic with them and explain them what is happening, what is going. And along this last year, we have a significant number of interactions with patients association, okay? And at the very beginning, you are thinking, okay, what I'm going to say to these guys, because you don't want to generate over expectations. So you have something which is super good, but it's curing a mice. And it's a mice model of the disease. And then you have fear to, what I'm doing. And the thing is that they understand perfectly what you are doing if you explain it in the simple words. So it's better that you tell to them that it appears in the news that you have something to cure Parkinson. This is something super bad for them. And this is what we try to make in the media. So we try to say, we are working for you. This is what we are doing. We are working for them. And we arrived until this point. We are going to do the best of our effort to push it there. But we don't want overstatements, because this creates false expectative for these people. And another thing which is nice is it's like a kind of global journal club. So essentially you bring there your papers or your thinkings, and you have discussions, and it's a way to disseminate also the paper. So essentially, I used in the past to send papers to my students. Okay, so I say, okay, this paper is super interesting. What I'm doing now is I post it, and then it's up to them to see that the post is there. And the journal clubs we make in the lab. In many cases, of the things we saw in the media. So we are looking still to PubMed, but I mean, sometimes it's easier to look just at X or like LinkedIn or Instagram, that we have not only the paper but a little explanation of the author on what he thinks or she thinks is discovered. So I think for me it's super important. And my people around, I mean, I am like not applying them, but let's say, making them to be attached to this way of disseminating science is part of our work.
Mm-hmm. Thank you so much for the discussion. It was very nice, and the idea that there are people that are feeling this in Italy is called third mission. The idea that this disseminating mission is part of the daily life of a scientist, it's reassuring. It's nice to think that there are people explaining their own job. Thank you for finding the time. And we hope to see you soon again in one of the next meetings.
Thanks a lot also for your time. This has been super nice, this interview.
Thank you.